And so a point I really want to hammer in is that single-cell RNA-seq data does not look like bulk RNA-seq data in a number of ways. And so just to kind of get back to this point about technical noise in single-cell data, I really want to harp on this point that single-cell RNA-seq data is very sparse. And so again, for some technologies like 10x, again, up to 95% of your table can be zero, right? And so essentially the number of transcripts detected is essentially much lower in these droplet-based single-cell RNA-seq methods compared to bulk RNA-seq. And so what the figure on the left is meant to show you is that essentially the number of genes detected, which is inversely related to the dropout rate, is essentially in part a function of the number of reads that you sequence in a sample, but it's also in part a function of this particular type of technology you use. And so you can see that, for example, given the same number of reads sampled, bulk RNA yields the kind of highest number of genes detected, whereas some of these single cell technologies basically have, like the single cell smarter technology, basically detect the fewest number of genes given the same number of reads sampled. And so the point here is that the dropout effect, as well as other technical factors, vary pretty widely between the different technologies. And so this is one consideration when you're trying to, for example, look at data from across multiple single cell platforms. And so the figure on the right here that's labeled 0 counts related to mean gene expression level is basically meant to illustrate what dropout looks like, both at the expression level for individual genes level is, and it's meant to illustrate the fact that this kind of zero inflation, or this feature that some genes are just never detected in some cells, actually varies depending on the mean expression level of that gene. And so in part A of this diagram, you can see that basically what's illustrated are the histograms of expression measurements for 3 different genes across a population of cells, and the genes are ordered from left to right in terms of mean gene expression level. And so you can see that on the left, for the gene BB510, basically this gene is on average poorly expressed overall. And so you can see a relatively big peak at 0, which means that there's a lot of cells in which this gene is expressed at it's detected at basically 0 counts. And that's in contrast to the gene C1orf86 shown on the far right, which of these 3 genes is the most highly expressed of all of them. And you can see the peak at 0 is relatively small compared to the other 2 genes. And so what this kind of illustrates to you is that the so-called dropout rate, or the fraction of cells in which a gene is detected, is not detected, is basically related to the average expression level of that gene. And so genes that are more highly expressed on average tend to be dropped out or tend to be measured at 0 expression less often than genes that are poorly expressed. And so this is another way of saying that this kind of dropout effect, where you don't see detection of a gene at all, occurs more frequently with genes that are poorly expressed than for genes that are highly expressed. And so despite all of these kind of technical factors of variation and also the biological factors of variation that drive variation in expression across single-cell RNA-seq data, by and large, single-cell RNA-seq measurements agree with bulk RNA-seq measurements when your library quality is good and your number of reads is high. And so here's a plot where on the y-axis represents the expression level of a gene measured by bulk RNA-seq, and the x-axis represents expression measured through the Fluidigm C1 platform. And so each point represents a single gene. And so you can basically see that on average, there's a relatively high correlation between bulk RNA-seq and Fluidigm C1. You can see there's a bit more noise at the lower end of expression, so on the bottom left, whereas more highly expressed genes in top right tend to be in more agreement between the two technologies. But on average, single-cell RNA-seq and bulk RNA-seq seem to correlate well when you have high read depth. So like I mentioned on the previous slide, the amount of noise in a gene expression measurement tends to be inversely related to the expression level of that gene. So here's a plot where the y-axis represents some measure of expression variability, in this case coefficient of variation, and the x-axis represents the average expression level of a gene, and each point in this plot represents a single gene. And so the main point here is that as the mean expression level of a gene increases from left to right, the amount of variability in the estimate of its expression measurement is decreasing. And so basically, poorly expressed genes are harder to estimate accurately. So what's also important to note is that the amount of noise in your expression measurements depends a lot on how you capture those RNAs. And so, for example, for UMI-based methods, like most of the droplet-based methods, you can see a relatively low amount of noise. So the y-axis here, the squared coefficient of variation, is yet another measure of variability in expression. And you can see that this squared coefficient of variation is the spread around the red line, which is the line of best fit, is relatively low and constant across the entire red line. Whereas for non-UMI based methods that don't, they can't detect PCR duplicates, for example, you can see that there's much wider spread in the squared coefficient of variation than compared to UMI based methods. And so once you've done some basic data pre-processing and calculated your table of quantifications of how many times each gene was observed in each cell in your dataset, you then typically take that table and then you punch it through any number of different workflows in order to perform tasks like trajectory inference or cell type identification that we talked about previously. And so here I'll basically just illustrate the basic steps of the single-cell RNA-seq workflow. And so again, you start out with your table of quantifications of each gene in each cell. And then the first step that is usually taken after quality control and things like this is you typically first see dimensionality reduction being applied. And so dimensionality reduction, in short, refers to techniques like principal components analysis that we talked about previously. And the main goal of dimensionality reduction, in my mind, is to remove essentially a lot of the noise in your data. And so again, we mentioned briefly that there's a lot of sources of variation in expression in single-cell RNA-seq datasets. And so the general line of thinking is that if you kind of only keep the first few principal components that correspond to your dataset, and you kind of throw away all of the other principal components in your dataset, then essentially you're just kind of removing a lot of the effects of those kind of other technical sources of variation in your data.